biogas production is basically it is kind of the clean energy, which is known as the mostly known as the bio CNG. You have heard about that, the different kind of the biogas used for the as a fuel. So we are also going to produce the biogas in lab, and how we will prepare the medium for that. Mostly it require an anaerobic condition in which we will do the work on the serum bottles, or maybe the closed conditions in which the anaerobic microbes grow and then produce the different kind of biogases. Mostly it contains the methane and the CO2 or the hydrogen we can produce like this in different kind of the microorganisms we can use and provide the condition medium and all. So we can produce the microorganisms have the ability to produce gas with that at the time of the anaerobic digestion or anaerobic fermentation, and mostly the specific medium used for this is the anaerobic evolved medium. Supplementations are required in this whole process, in which the vitamins and trace elements and mostly the major carbon source is given as a sodium acetate, in which it is required as a kind of the acidogenesis to the solvagenesis process, in which a two-stage process solves for the production of the gases out of it. And we can use the different kind of the anaerobic substrate or the inoculums, like the wastewater sludge are also a kind of the a source of the anaerobic microbes that are going to be the inoculum for the production of the biogas. And different kind of substrates are used, like the food waste in labs, like the kitchen waste are mostly used for the production of biogas. And somehow you have heard about the cow dung is used for the biogas production in different areas where the biogas plants are established, and many of the petroleum companies are working on just replacing the petrol fuel to allow the biofuel, and bio CNG is now coming in this existence in the current scenario, and this is also having a mixture of this carbon nitrogen gases, and mostly we can use the methane for that is also used in the households, like the liquid petroleum gas, and is similar to like this. We can use the biomethane for making the kitchen making the kitchen food items for using as a burning fuel for this in your cylinder or something. And so the pipeline you can transfer, you can fit up in the kitchen, and then you can use it as a fuel, and this is working as the same LPG. And this is a kind of the clean energy, and it can be produced by the microorganisms, and when the conditions are provided very much high, and we will prepare this kind of the conditions in serum bottles, and we will see how the gas is we will measure the gas by the inverted water the standard method, and which we can calculate how much of the ml or the liters of the gas is produced by the whole setup you have done with the 100 ml or the 50 ml, or maybe the higher volume of your substrate volume medium preparation for the fermentation is given. And let's start firstly preparing of the medium for it, and then we'll prepare the medium, then autoclave it, and then we will just add the inoculum taken from the sludge or the wastewater. Last time we have also prepared this kind of the sludge sample, and then we will inoculate it and keep it of the incubation. After the incubation, we will see how the gas is producing and how much it is there. We will measure how much it is there, and this can be analyzed by the different kind of the gas analyzers, like the GC chromatography, in which we use mostly in the industry the RG refinery gas analyzer is used, in which you just inject this kind of the GC. We just inject the gas sample, and then you can easily identify that which kind of the percentage of the CO2 or the methane are there, or other hydrogen is there. So this is let's start with this firstly preparing medium, and then do the fermentation and the identification of the biogas production. Now you can see the resin showing the color as a dark red pinkish red in color. So it is showing the condition anaerobic. When we provide the condition in anaerobic condition by purging CO2, it should become the colorless, and it is showing that the condition is totally anaerobic. So we will purge the CO2 here, and then we will see the color. You can see. When we provide the anaerobic condition here, now you can see the color is changed now. Now you can see when we have provided the CO2, it is showing the colorless, transparent, totally transparent. It is little, the media color is coming, but the resin color goes down, goes to colorless. So it means that the condition is now fully anaerobic. Now we can inoculate the culture after autoclaving and all the process. So this is how we can measure how the condition is anaerobic or not in this condition. Like you can provide the indicator like resin, and that will help you to maintain the condition anaerobic all the time. And kind of the oxygen or something will come, it will show the color change. So this kind of the anaerobic medium is going to use for the for adding up the inoculum and then generation of the biogas anaerobic conditions over there. See, we are now going to start the CO2 purging by this line, and all are in red in color, the serum bottles we have prepared. You can see. So we will take one and then start. Actually we will put one to CO2 out and one is the CO2 in now. We will check the CO2 is coming out or not like this. You can see there is a bubble. Right now it is showing pink in color because the resin showing the indication of the oxygen. When there is a condition become totally anaerobic, then it will show as a disappearance of the color. Now you can see the color disappearance starts now. It will retain the media color again. Now you can see how it is changed and the total transparency of the color. And you can compare the aerobic and anaerobic color. This. Similarly, we will prepare all the serum bottles and then do the autoclaving of it, and then after the autoclave we will just inoculate the inoculum and then go set for the incubation and see how the gas is produced by them. You can see we have done the purging with all the samples, and then you can see now there is no color change after the mixing and all. This means the condition is fully anaerobic, and we are just completing last one, and it is also showing the disappearance of the color. totally disappeared now. After that we will do the autoclaving. Cleaning is over. Now we are removing it. Learn how to set up the gas analyzation by inverted cylinder. and we will see that how to use it. This is the silicone tubing, we are going to use it, and these are the needles to pass the gas from this. Firstly we will fill the milling cylinder, the water to the milling cylinder. Okay, now we will set up. Now we have to flip it. Then we will see the marking, and it is showing how much, so it is showing around like the inverted one at this point. We will mark it here. Now we will check the gas. So this is the whole setup we have already done with this serum bottle, and then the inoculation and all the generation of the biogas. Now we are going to check it. So we will just add in this and find out how much it is there. And how the gas is coming out of it, and we are inserting. Continuously you can see. You have learned about that how we have prepared the biogas in the lab, and how like we have prepared the medium anaerobic condition provided for that, and we have just used the indicator to find out the anaerobic conditions are okay or not, and then we have inoculated the waste sludge inoculum, and having the anaerobic microbes for the community over there. And we have seen that how we have just do the setup of the to find out the gas through the milling cylinder inverted one, and we have seen that the production of the biogas, like mostly it is the methane gas, which is mostly used as a biogas in the cylinders for a kind of the gas to energy to be provided for the vehicles to work to run, like the bio CNG, and this is basically the pure methane is used, and you have produced, which comes with the CO2 H2 gas or other gas will come. So we have to purify the one out of it from the raw raw gas, total raw gas, and then the pure biomethane, which can be used for the as a petroleum fuel for running of the vehicle or in the kitchen supplies and other things. So similarly, it is a small setup we have used. So we can do the large reactors like anaerobic digesters, and we can use the municipal large digestions for this, and like the cow dung digestions for production of biogas currently used in the rural areas, and very large plants for the or scale up scale up of the plant at the large scale in a million tons to produce the biogas in tons, and commercially supply the biogas for the as a clean energy to the to running to provide filling stations for filling up like the CNG stations, pump and all. So for providing the cylinder filling for the vehicles, and it is good and clean energy source, and sustainably it is also producing the fertilizer organic waste that can be used as fertilizer for the growth of the like for the organic farming and the things like you have to which is reaching in the organic substrate, and also it is utilizing and treating the sludge also, and the remaining waste solid sludge will left over there only, and it is also reducing many of the environmental problems when there is a landfill of the waste is like from the paper pulp industry the waste is there, and some other municipal waste just openly fell down dumped over the landfill, so it produces the gases and causes the environmental problems. So to reduce these kinds of the use these waste like kitchen waste, food waste, municipal solid waste, rubber waste or other waste similarly which have the ability to be digested by the anaerobic microbes, and then we can easily produce the biogas at a large scale, and where it commercially can be it can be go for the commercial production of it, and then can be sold as a biofuel for the transport and other services. Okay, thank you.